Hello and welcome to the review video for Genetics Unit 5. We are talking about quantitative genetics. So quantitative traits are continuous, while qualitative traits are discontinuous. Discontinuous traits mean they have distinct phenotypes and there's usually just one or two. So that would be something like um, if per se, there's only two types of feathers, long feathers or short feathers. Do the chickens have long feathers or short feathers? That would be a discontinuous trait. Quantitative trait is actually what feathers are. In reality is there's a wide range of feathers. Feathers can be anything from really long to really short. Quantitative traits are something that can usually be described in a normal distribution. So there's a very wide range of phenotypes, kind of like what you see for IQ, height, stuff like that. For quantitative gene traits, the genotype of these can be due to a lot of different things. We have polygenic, multiallelic. We talked about these in past units. And it's pretty hard to predict genotype based on the phenotype because of all of these things, plus gene interaction, and just how complex these traits usually are. We have different kinds, and for meristic, this is kind of like what we're talking about with immunity, where there is technically a finite number of combinations you can make, but that is a very large number. We also have threshold traits, which are two distinct phenotypes, but the underlying cause has continuous variability, meaning that the underlying cause could be completely different from person to person, but the phenotypes would be the same and in two distinct phenotypes. We also have the additive effect of alleles. So we have major alleles add to the dominance, to the expression, whereas minor alleles don't do anything. So if you had three major alleles signified by big case letters and three minor alleles, then you would have, say, that's average. Say that means you are of an average height. But then if you had, out of those six alleles, if you had five of them were major and only one of them was minor, then you would be a very tall person. Whereas if four of them were minor and only two of them were major, you would be a very short person. That's kind of how additive alleles work. The more loci involved in a trait, the greater the phenotypic range, because then you just have more genes in play. Let's have a quick statistics lesson here with calculating probability of an offspring having the same phenotype as a parent. So this calculation is 1 over 4 to the n. So basically, n is the number of loci. So to calculate the probability that the offspring will have the same genotype as the parent. Say if there are 3 loci, it would be 1 over 4 to the 3, which equals 0.0156. So that would be 1.56% chance that this offspring would have the same phenotype as the parent. These continuous traits usually show normal distributions. Normal distributions are shaped like bell curves, and this means that in the middle, that's average, that's what the most people have, and at these two ends are the extremes. So if this is super short, and this is really tall, then the middle would be average. And most people would have this middle average, even though this is short and this is tall, most people fall under this average. There's not a lot of people who fall under these two ends of the bell curve. So we have a large range of how to measure this. We have mean, which is the average of the population. We have variance, which is the average squared deviance from the mean, and standard deviation, which is the square root of variance. These are connected. Standard deviation is how far away from the mean are we? Um, like in a bell curve, we usually have up to 3 standard deviations. So are we 1 standard deviation away? Are we 2 standard deviations away? The more standard deviations you are away from the mean is the more unusual or rare or uncommon this is to appear or happen. If a peak is skewed, that means that the peak is not centered on the center. If the center was here, but the peak is here, that would mean it's skewed to the right. This is skewed to the right. It's a little weird, but you could think of wherever the tail is, that's where it's skewed towards. It's not skewed towards the left, it's skewed to the right, and vice versa. We also have bimodality, which would mean there are multiple peaks in this graph, which instead of just one normal distribution, there's actually a few peaks. So in this bimodal graph, basically what we're seeing is that even though the traits vary as you go up, it's not necessarily farther away you are from the mean, the less common it is. It means that these traits are most common at the peaks and less common in the valleys. So if this was height again, this would be saying, and this is short, and this is tall, this would be saying that kind of short people are very common, medium people are common, and kind of tall people are common, but not really in these in between. So if this was, say, like 5'4", 5'8", and 5, uh, or let's do 6 foot, 6 foot. Basically, we're saying that there are a lot of people that are 5'4", a lot of people that are 5'8", a lot of people that are 6 foot, but there's not a lot in between. So there's not a lot of people that are, say, 5'10", 5'11". There are not a lot of people that are 5 foot flat. Most people fall under one of these peaks, and not a lot of people fall under anything else. We also have line graphs. So if we have a line graph, we usually have a bunch of dots, and we form a line around these dots. It's called the line of best fit. So if we have a line that's going up towards the right corner, that is a positive correlation. If we have a line that is going down towards the right side, it's a negative correlation. If the dots are quite close to the line, they follow the line really closely, that would be a strong correlation. If they were really spread out and it kind of didn't really make a lot of sense, that would be a weak correlation. We also have something called a regression coefficient, and that's symbolized by R. Regression coefficient has a range of -1 to 1, so you'll never find any values outside of -1 and 1. -1 means a really strong negative correlation. +1 means a really strong positive correlation. Closer you get to 0 on either side means a weaker correlation. So a score of -0.2 would indicate a very weak negative correlation, whereas a score of, say, 0.1 would indicate a weak positive correlation. So that's how regression coefficient works. We also talk about inbreeding, which inbreeding means they're homozygous at most loci, and that just means that it's clear that there's not a lot of genetic variation in the population. Most of them are genetically very similar, which is usually a bad thing. We have the heritability of quantitative traits. And these are a bunch of variables and equations. So make sure that you take note of these equations here and what they mean, and we will use them in calculations. Say you might be given genetic variability, environmental variability, and gene interaction, environment or interaction, and asked to calculate the population variance. We have such thing called broad sense heritability, which is symbolized by a capital H squared, and that is the proportion of phenotypic variance due to genetic variability. Basically, how much is the difference in phenotypes due to genetics, not environment. So if H squared is 0, that means it's all environmental that is influencing this phenotype. If H squared equals 1, that means that's all genetic factors influencing these phenotypic differences. Narrow sense heritability, h lowercase h squared, is the proportion of variance due to additive genetic variability. So how likely are the offspring to resemble their parents, basically? And a reminder is heritability is a statistic, and it only indicates how much variability in a trait has a genetic component. It says nothing about that trait itself, or whether which phenotype is recessive or which phenotype is the dominant. It just says that this trait is largely influenced by genetics or largely influenced by environment. If you are asked to find the narrow sense heritability from a line of best fit graph. If you will have a slope of this line, and the equation of the line is in the form y equals bx plus a, or mx plus b, if that's something that you're more familiar with. But basically, uh, our narrow sense heritability is this slope. So it is b. So if the equation of this line was y equals 0.5x plus 5, our narrow sense heritability would be this 0.5. Some notes on heritability: it does not apply to individuals, only populations. And while it does apply to populations, it is not universal. This is population dependent. Even if heritability is really high, that means that that does not mean that the environment cannot influence the trait. The environment still can influence the trait. And it also does not indicate anything about, again, recessive dominant phenotypes. It just indicates if that trait corresponds to a high genetic factor or environmental factor. We have the selection of quantitative traits, and we use that in agricultural production. And basically, when we do natural selection, we try to push, say, if we want larger ears of corn, we try to push the average phenotype towards a larger ear rather than a smaller ear by selective breeding. And narrow sense heritability can actually tell us how successful our our artificial selection will be. Because if we have a really high um, lowercase h squared, that would indicate that offspring are really likely to resemble their parents, meaning that the artificial selection will likely be really successful. But if we have a low narrow sense heritability, that means that the offspring don't really have as much of a chance to resemble their parents. And that kind of means that even if you cross two really, really good parents together, their children might not actually reflect that. It's kind of hard to think of an example of this, but all I can give is a really weird example, as I'm known for. Minecraft horses, their parents have kind of a low, narrow sense heritability because when they have children, actually their statistics tend to go towards an average um, total horse speed instead of average of the parents. It kind of skews back towards the mean instead of getting better. So that would be an example of low narrow sense heritability. That would mean that even if you bred these two really fast horses together, you might not produce a really fast baby because that trait is not highly influenced by genetics. It's influenced by the environment, or in this case, the game's coding. So that is kind of a lesson in narrow sense heritability and how we can determine the successfulness of artificial selection via that number. We also have the selection differential, which is represented by S. And that is how different is the phenotype of the selected parents different from the mean of population. It basically says how above or below average are you. So we calculate this by the mean of parents minus the mean of the population. And we can actually use this in combination with our narrow sense heritability to find what the offspring's average of this trait will be. So if we are looking at, again, corn, if we're looking at the size of the corn ears, and if we have a high narrow sense heritability and the parents are very above average, so say if we have a high narrow sense heritability of, say, our H equals 0.8, that's pretty high. And then we have our S. So let's say the parents are 3 inches larger than normal. If we multiply these, 0.8 times 3, we get 2.4. We can then add that to the average. Let's say the average corn ear length is 7 inches. 7 inches plus 2.4 equals the average of the offspring. So the offspring of these two parents that have corn ears 3 inches larger than average would produce offspring with an average corn ear length of 9.4. So that's how we use um, response to selection to determine what the average of the offspring would be when compared to their parents, and we have that narrow sense heritability brought in. So if we did this and we had a really small h, if we had, say, h was 0.2, so there wasn't a huge genetic component to this corn ear trait, and we still had s equals 3 inches. So then if we did this, it would be 0.2 times 3, which equals 0.6. So then 7 plus 0.6, because 7 is our average, would be 7.6. So the average corn ear length of the offspring would be 7.6. So you can see how our narrow sense heritability really impacts how the offspring will turn out from these cross of the two parents that might both be good things that we were really looking for, but it depends on the narrow sense heritability of whether that will actually pass on to the children or not. We have correlated responses. So many traits are correlated due to genetics or environmental conditions, which can limit the selection potential. That's like if corn ears, corn ear length is correlated with root length of the corn because I don't know, larger roots equal more nutrients or something like that. This is all made up. But if that's so, and we don't realize that, if we crossed two larger corn ears, but they had shorter roots, and we started getting um, offspring with shorter corn ears, we would be like, what's happening here? I don't know. Maybe it just has a really low narrow sense heritability. But actually, what in fact is, we would need to have two large corn ears and large roots for them to actually pass that trait of large corn ears on effectively. We can also map quantitative traits using quantitative trait locuses, QTLs, and this is the chromosomal region that can be associated with inheritance of the trait. It is the region, not a gene, and can be mapped using linkage and genetic markers like we talked about in the last unit. Okay, we're also going to talk about population genetics. So, population genetics, we have a gene pool, and that is the collection of genes or alleles present in the population that contribute to the phenotype. So, this is basically all the genes and alleles that can make up the phenotype in a location or a population. And so that kind of contributes to different gene pools, mean that in different locations, the same species can have a different phenotype. It's kind of shown in like the insane variation of human phenotypes. And so in different regions of the world, humans have a very different phenotype, and that's because of different gene pools. We have genotypic frequency. So that is the frequency of genes or alleles in the population. And how we calculate that is the sum of the particular genotype divided by the total number of individuals in the sample. So this is an example right here. And we have different numbers of individuals with a genotype, and we have the total, and then we just divide them out, and those would be our genotype frequencies. So that would be the frequency of the number of people with an AA, so all dominant genotype, would be 0.25, which means that in this population, roughly 1/4 of the people would have the genotype capital A, capital A. We can do the same. We can actually also figure out the allelic frequencies. So that's the frequency of this specific allele. This is not linked to the phenotype, whereas this is a little more linked to the phenotype because it's the genotype. But this is the frequency of the allele. We're talking about the frequency of capital A versus lowercase a. So basically, we can mainly look at these equations down here, and that is the frequency plus the frequency of capital AA in P's case, plus 1/2 the frequency of capital A lowercase a. So if we were looking back at this example, we would have P equals the frequency of capital AA, which is 0.25, plus 1/2 the frequency of capital A lowercase a, which is 0.508. And this would equal 0.25 plus 1/2 times 0.508 equals 0.504. That would be our P. And now one thing we can know is, according to Hardy-Weinberg's law, P plus Q equals 1. So then, using algebra, we can have Q equals 1 minus P. So therefore, in this example, Q would equal 0.496. And that takes us straight into Hardy-Weinberg's law, which is basically these set of equations. Now, Hardy-Weinberg's law operates on a set of very defined assumptions, and these assumptions are that the population is infinitely large, random mating occurs, so no natural selection, it's not affected by mutation or migration, and reproduction does not cause changes in allele frequency. So basically, it assumes that allele frequencies in a population are fixed and unchanging. And with this assumption, we can use these equations to calculate allele frequencies. So basically, P plus Q equals 1, and all of these other equations, all of which you should definitely know. These implications of Hardy-Weinberg, which means that at Hardy-Weinberg equilibrium, as this state of assumptions is called, allele frequencies will not change and no evolution will occur. Additionally, these allele frequencies can be used to determine the genotypic frequencies. Frequency of heterozygous zygotes is highest when the allele frequencies are between 0.33 and 0.66. Basically, they're the absolute highest when P equals Q, which is equal to 0. When both of them are equal to 0.5. This law can apply to loci with 3 alleles and X-linked traits, but it's kind of limited beyond that. We have a quick example, which is how we could find the other frequencies based on just one. So we have a recessive frequency, and then we find the Q from our Q squared, and then we use 1 equals Q plus P to find P, and then P squared would be the frequency of capital A, and then 2PQ would be the frequency of capital A, lowercase a, and we already have the frequency of lowercase a. So this is how we can calculate genotypic frequencies using these Hardy-Weinberg equations. And a second reminder, this only applies at equilibrium. How to determine if this equilibrium actually applies is we look at our observed, calculate the frequencies of our observed, and then we use these Hardy-Weinberg laws to calculate the expected and compare them using a chi-square test and basically make sure that the results are not statistically significant, meaning we do not want to reject the null hypothesis if we want to assume Hardy-Weinberg equilibrium. There are a lot of effects on genotypic frequency that are outside of Hardy-Weinberg equilibrium, such as mating effects. We have associative mating, which is preference for mating partners. So positive assortative mating would be picking partners that look similar to themselves. Negative would be picking partners that look different from themselves. We also have inbreeding, which increases homozygosity, as I mentioned earlier. We have F, which is the inbreeding coefficient, and the higher the number of F, the closer the population is related, and the faster it will reach complete homozygosity, meaning everybody basically has the same genome. And this usually leads to negative effects from the population because it makes the population harder to evolve, harder to use natural selection to overcome barriers such as disease or like a natural disaster or something like that. It can also cause issues because recessive alleles that shouldn't be popping up sometimes get more expressed, and that's why, again, it's really usually bad to inbreed organisms because then you could come up with all sorts of defects and not good stuff. And we have a factor called hybrid vigor, and that means you have a low F, and that means you have less inbreeding, and that means the fitness of the population is usually going. Although it is worth it to note, a lot of more simpler organisms like slugs can actually thrive off of inbreeding. It's not exactly terribly detrimental to less complex organisms. We also have the effect of mutation on genotypic frequencies, And when we think of mutation in regards to genotypic frequencies, that just means that one P allele, dominant allele, is transformed into one Q allele, the recessive allele. And usually in Hardy-Weinberg equilibrium, the rate of the forward and reverse mutations are similar enough that they cancel each other out, basically. So every time a mutation happens that transfers one P allele to a Q allele, a reverse mutation happens that transfers a Q allele to a P allele, so everything ends up balancing out. Mutation rates are usually pretty low, mutations that are passed on, but over a long time, a period of time, they can be significant, which is how we can kind of get natural selection and evolution. We also have effects of migration, and basically, if you have migration between populations with different allelic frequencies, the mixing can change these frequencies, and basically, it will make the populations more similar because there's increased genetic variation and the variation crosses over. So then, slowly, both populations have the genetic variation of those two different populations. So they have a bunch of genetic variation and they're actually more genetically similar. We have the calculation for variance that uses our Hardy-Weinberg. N equals the number of people in that population. We also have genetic drift, which is that causes allele frequencies to be fixated to one allele or the other. So say, oh, one, the dominant allele, or it could be fixated to the recessive allele. And this leads to a loss of variation. And actually, it can be how populations diverge because if one population is fixated on one allele and another population is fixated on another allele, they might actually diverge enough to the point where they become completely new species because they are reproductively isolated, which we will get to later when we're about to talk about evolution. But for now, genetic drift is basically the favoring and fixation of one allele over the other. We also have the founder effect, which means if, say, a few individuals from a population are stranded on an island and they start to build their own population of that organism, that population will only contain as much variation as the original few parents did, and that can create less genetic variation to be had. We also have the bottleneck effect, which is basically if there's a huge natural disaster, so natural selection is not involved. This is like a fire, a hurricane, something catastrophic, and the population size is heavily shrunken down. And we have to rebuild that population. A lot of times, we will only have the genetic variation available to us of those original members that start to rebuild the population. This is a huge problem with trying to bring species that are close to extinction back from extinction because when you're working with only like 50 surviving individuals, it's really hard to have genetic variation in that population. And sometimes populations can crash because there's not enough genetic variation in these newly revived populations. And so they're more susceptible to disease or deformations and stuff like that. We also have natural selection and mutation. So, natural selection often removes negative alleles from the population, and that can usually balance out negative mutations that occur. Of course, then we talk about in different settings where that doesn't happen. For example, in like breeding dogs, a lot of times issues that would be deadly for dogs in the wild aren't as bad as issues when they're kept in captivity. So we sometimes end with a lot of dogs that are bred specifically for their looks, but that look can have a lot of problems that probably wouldn't survive in the wild, but can be taken care of. That's an example of natural selection not occurring and balancing out those negative mutations. We also have natural selection and fitness. And so basically, when natural selection does work, it often selects for the positive alleles that make the species as a whole stronger and more able to survive and reproduce. And basically, fitness is a percentage of offspring produced by a certain genotype that is compared to the most prolific genotype, and we kind of can measure that. And there are a few slides in the slide deck talking about fitness. and how to calculate that, but that will not really be tested on. But you do need to know like what fitness is and the overall measure of fitness. Okay, and now we're going to talk about evolutionary genetics. So, evolutionary genetics, we have evolution, which basically means that all the stuff we were talking about just a bit second ago means the population is not in Hardy-Weinberg equilibrium. And basically that the population is changing and that the genetic makeup of the population is changing. We have two ways that evolution is really marked. We have it by cladogenesis, which is basically splitting of one lineage into two, or splitting one species into two. And we have anagenesis, which is evolution within a lineage with the passage of time. So basically, this means that if we say we have our phylogenetic tree, if we have a branch, that would be cladogenesis. But this getting from point A to point B, all of this in between is also evolution. It's just anagenesis, not cladogenesis. So it means that we still have the same species and they're still evolving. It just hasn't diverged into two different species. And evolution mainly depends on phenotypic variation, actually, because evolution is based heavily on natural selection, because this phenotype will determine if the individual is more successful in reproducing or not. And basically, if you reproduce, then your genetics are passed on, etc., and therefore, then the population slowly shifts towards that genetic variation or that genotype if it's reproductively advantageous. Um, so we have molecular evolution, which is basically looking at the DNA of different genes between different species, comparing them, and seeing how exactly the DNA evolved over time. And this is really interesting, but it also doesn't necessarily reflect the phenotype, which is the main driving factor for evolution. We have different ways that this can happen. We have DNA sequence variation, which is variance in the DNA, obviously. We have this can cause variations in RFLPs or short tandem repeats, what we were talking about last unit. And basically, that's a way that researchers can quantify these genetic changes. We also have variation in protein sequences, which, as we know, silent mutations don't affect the actual protein. So, this can not sometimes not reflect the true genetic variability, but it does show how certain genetic variability does affect or does not affect the phenotype. Biological distinction of a species is just a species is a species when they can breed with each other and produce viable offspring that are able to continue to reproduce. If these two organisms can breed and produce viable offspring that can reproduce. So that doesn't mean they can't be sterile. So, like if we cross a tiger and a lion, we can get a liger, but this liger is sterile. So it's not a valid species. It's a hybrid. But as long as these two can breed naturally and produce viable, fertile offspring, that is, they are a species. Once they start not being able to breed with another organism, then they are classified as different species. And this is called reproductive isolation, where basically these organisms can no longer mate with anybody but organisms of the same species, which means they're reproductively isolated from the species around them, and so therefore they're now considered their own distinct species. And this can actually happen in a variety of ways. We have allopatric speciation, and that just means that these two populations were geographically separated by like a mountain or an ocean or something, and they could not access each other. And because they were separated for so long, they evolved away from each other. They evolved so to be so different from each other that they could no longer reproduce and produce healthy, viable, fertile offspring. And that means that they are now two different species. So allopatric speciation arises when populations are isolated from each other by geography. We also have sympatric speciation, where species arise in the same area, but they are usually reproductively isolated um, for some other reason. Say, maybe physiologically they're reproductively isolated, or whenever they try to have children, they're not viable. So basically, they're reproductively isolated some other way. Polyploidy organisms, this can happen in a lot of plants, is when two plants try to reproduce that are not the same species. Difference in ploidy levels can create offspring that may be viable, but they may not be able to mate with the same species as their parents. So this can also create kind of a different reproductive isolation where this hybrid, it can still reproduce, but it cannot reproduce with its parent species. And this is when we're talking about polyploids, which we've talked about in previous units. Um, a mutation in the molecular clock. So basically, we assume that the rate of change in any gene or protein is constant, and basically, the rate of this change or mutation between two species can provide the rate of evolution. And we can also see that changes in regulatory regions, changes in base pairs in regulatory regions, can also influence evolution because it influences gene expression. There's also evolution at the genome level. Sometimes exons can be shuffled, which creates different protein sequences, and or gene duplication or whole genome duplication can either increase ploidy levels or it can have some gene dosage effects that, if they aren't lethal, can actually be something of a way for speciation to occur. Genetic aspects of development. So basically, the potency or the ability of like a stem cell to develop into different kinds of cells is really important. And how that happens, if there are things affected that say affect the ability of the cell to create, and if something happens that affects the ability of a stem cell to differentiate, that can affect the organism as a whole. And this can also be a huge thing in development because if stem cells differentiate in the wrong order, say if they develop the organs out of order, this can also really change how the organism develops. And this is controlled by a lot of genetic factors, stuff like gene expression, etc. We also have epigenetics and regulation of cell development can also affect this. We also have the genetics of pattern formation. So basically, how the body develops, we have different kind of polarities. So we know which is our feet and which is our head when you're developing as an egg. From like an egg all the way to an embryo to like a full-fledged organism, you have to know which part side of the egg starts at the head, which is the tail. And basically, that is actually determined by genetics, which is really cool. And so we have these things called egg polarity genes that are expressed in the developing egg from the maternal genes that basically determine the polarity of the developing offspring. And there's a protein gradient that establishes this polarity. We also have such thing as Hox genes, which are basically associated with the development of organisms. So basically, if there's more Hox genes, then this actually has more developmental complexity. So for example, humans would have more Hox genes than, say, I don't know, an isopod or a roly-poly or an insect. We have cancer genetics. A quick little segment on cancer genetics. And basically, when cancer arises, it's usually due to a mutation to a gene that controls proper cell growth and differentiation. And most of these mutations are somatic, so not sex cells. And so cancer is not an inheritable disease. Although there are genetic factors that can increase the risk of cancer, it's not necessarily or at all inheritable. Um, some mutations can be present in sex cells, and that again increases the likelihood that cancer will develop, but usually doesn't actually lead to actual cancer. And cancer is also an accumulation of mutations. It is not just one mutation, one and done immediately triggers cancer, but a lot of things have to break in order for cancer to occur. And usually, one of a few of these cancer, quote unquote, cancer causing genes have to start functioning irregularly before cancer actually starts. So, cancer genes like oncogenes or proto-oncogenes, so that's cyclins or CDKs, and tumor suppressor genes are affected. So basically, if you could think about it, if a tumor suppressor gene is broken, so it's no longer suppressing tumors, then now this cell perhaps wants to create a tumor because that gene is now broken. And also, other cancer related genes can be DNA repair genes, telomerase, which prevents aging, which is why as you age, you're more likely to have cancer, and cell signaling pathways. So, all of these can play a huge role in cancer, but it's often a lot more complex in returns to the genetic element than we really think because we usually we're like, oh, it's this cancer. Okay, they must have a mutation. But actually, it's a variety of mutations that all built up in one place in a really bad sequence to create cancer. So yes, that is our Unit 5 review video. That will be the last video for genetics that I will be making, and I hope you all have a great day and a good time studying.